Welcome to the Explainer. If you work in tech, you already know the absolute golden rule of shipping a new product, right? You don't just guess what's going to work. You test it. You run an A/B test. Well, it turns out medicine has been doing this exact same thing for decades. The only difference? Their platform is human biology, and, the stakes are quite a bit higher than a drop in click-through rates. Today, we're taking a really fascinating look at Lesson 3 from Evidence-Based Medicine for Techies. We're going to learn how to critically appraise a randomized controlled trial, or RCT. Think of it as learning how to read the ultimate biological A/B test. We're going to strip away all that dense medical jargon and just look at the raw logic of how clinical evidence is actually evaluated.
Okay, let's dive right into this. We've got a really clear, four-step roadmap today for reverse engineering a clinical trial. First, the ultimate A/B test. Then, the three-question framework. After that, we'll be crunching the trial results, and finally, your patient and applicability.
All right, starting off with Section 1, the ultimate A/B test. This is basically medicine's version of shipping to production. So let's look at how these two worlds actually collide. In the tech world, if you want to know, say, if a blue button works better than a red button, you randomize your user base, right? Half get the blue, half get the red, and boom, you measure the conversion rate. Well, in evidence-based medicine, an RCT does the exact same thing. You take a group of patients and randomly assign them into two cohorts. One gets the standard treatment or maybe a placebo, and that's your control group. The other gets the fancy new drug or intervention, which is your treatment group. From there, you just track the outcomes to see if the new feature actually improves the patient's biological metrics. It is literally the exact same data-driven mindset, just applied to human health instead of software. Moving on to Section 2, the three-question framework. Consider this your core algorithm for reading trials.
Now, if an RCT is our biological A/B test, then this framework is the core algorithm you need to actually read and verify those results. When a good clinician reads a trial, they don't just skip to the conclusion at the end of the paper. Honestly, that's a rookie mistake. Instead, they run it through a QA testing protocol. 3 essential steps. Step 1 is validity. Basically, is the data clean? Did they randomize properly? Were the patients blinded? Because look, if the study design is flawed from the get-go, the data is just garbage. Step 2 is results. Assuming the trial is valid, what actually happened? What's the size of the treatment effect? And exactly how precise is that measurement? And finally, Step 3, which is applicability. This is that absolutely crucial leap from theory to practice. It asks, can I actually apply these results to the specific breathing patient sitting right in front of me? This 3-step blueprint is how you systematically dismantle and understand literally any clinical trial.
Okay, let's jump into Section 3, crunching the trial results. This is the math of medicine, and it's where things get really interesting. Let's actually dig a little deeper into that second step of the framework, the results. Because this is where we get to turn those abstract clinical outcomes into concrete, verifiable numbers. We're looking at two very different ways of expressing the exact same result, absolute risk reduction, or ARR, versus relative risk reduction, or RRR. Let's paint a picture here. Say a certain disease kills 2 out of 100 people. A new drug comes along, and now only 1 out of 100 people die. The absolute risk reduction, the actual on-the-ground reality of the change in the population, is just 1%. The absolute baseline risk dropped from 2% down to 1%. It's a tangible but pretty small shift. But the relative risk reduction? Well, that compares the 1% difference against the original 2% baseline. So relatively speaking, the risk was cut in half. That is a 50% relative risk reduction. Do you see how sneaky that is? Pharmaceutical companies absolutely love relative risk reduction because, let's be honest, 50% lower risk sounds incredible on a billboard. It makes for fantastic marketing. But as analytical thinkers evaluating evidence, we have to look at the absolute risk reduction. We need to know what's actually happening in reality for a population, not just what looks good in a press release.
And that leads us perfectly to what might just be the most practical metric in all of evidence-based medicine, the NNT, or number needed to treat. Simply put, this is the number of patients you need to give a drug to in order to prevent exactly one additional bad outcome. Think about it. If a drug magically cures absolutely everyone who takes it, the NNT is 1. 1 treatment equals 1 cure. Simple. But here's the catch. Most drugs don't work like that at all. So I want you to think about this for a second. Why do you think this specific, slightly clunky metric matters so much more than some flashy percentage when a doctor and a patient are actually sitting down to make a real-world decision? Well, this brilliantly illustrates exactly why. From the source material, we see this scenario where a trial boasts a massive 40% risk reduction. Sounds like an absolute miracle drug, right? Sign me up. But when you actually sit down and calculate the number needed to treat, the NNT is 25. That means a doctor has to prescribe this drug to 25 different people, risking side effects, spending money, managing logistics for all 25 of them, just so one single person actually gets the benefit. The other 24 people, they get zero benefit. Nada. When you frame it as treating 25 people just to help one, the true utility of the intervention becomes instantly, strikingly clear. It completely strips away the marketing spin and hands you the raw, operational reality of the treatment.
But here's the thing. Crunching the math is really only half the battle. At the end of the day, we have to ask, does this specific patient even fit the trial average? This is where we have to pivot away from the raw spreadsheets and finally introduce the human element.
Which brings us to our final section, Section 4, Your Patient and Applicability. Welcome to the ultimate edge case.
You know, in software development, you always have to worry about the edge cases, right? Those weird users whose custom setups don't perfectly match your pristine, clean testing environment. Well, in medicine, literally every single patient is an edge case. Just because a drug reached statistical significance in a heavily monitored trial does not mean it seamlessly ships to production for every specific user. So the crucial point here is how we handle uncertainty. Clinical trials don't just hand us a single perfect number. They give us confidence intervals. A confidence interval represents a range where the true effect likely lives. It's essentially a mathematical admission of uncertainty. But honestly, the bigger challenge is the massive difference between averages and unique patients. An RCT tells you how a drug performed on average across thousands of carefully selected, highly controlled participants. But that patient sitting in the clinic, they might be older or taking 3 other random medications, or they have a completely different genetic background than the trial cohort. Clinical decision-making is this incredibly complex art of reconciling that clean, average trial data with the very messy, highly unique biological system of the individual sitting right there on the exam table. You're constantly synthesizing raw statistics with the nuanced reality of human biology.
Which leaves us with this final, really fundamental question to ponder. If the math works on average, how do we prove it works for this unique, de-identified patient? Reading an RCT, understanding absolute risk reduction, calculating the number needed to treat, these are incredibly powerful tools to have in your analytical toolkit. But we have to remember, they are just tools. True evidence-based medicine means taking that data and translating it to the individual. It requires you to look beyond the p-value and actually understand the human being. So the next time you see a flashy medical headline claiming some massive game-changing breakthrough, I challenge you to pause and ask, what's the absolute reduction? What is the NNT? And most importantly, does it actually apply to me? Thanks for joining me on this explainer, and hey, keep questioning the data.